So this lesson is on the prokaryotes, which is chapter 20.2. And we've now learned that there's 3 domains of life. 2 of them consist of prokaryotes, both bacteria and archaea. So this is kind of just a background on prokaryotes in general and what you should know about them. So prokaryotes are the fully functioning single-celled organisms of bacteria and archaea. Your textbook says that, and what that means is that just because they're prokaryotes or came before eukaryotes, or however you want to think about them, they are still cells. They perform all of the functions of life. They are alive. They can do everything they need to be alive. But they're all single-celled, like bacteria and archaea. They were discovered in the 17th century by Antoine van Leeuwenhoek. You guys might remember him. We talked about him in regular biology, or you would have. And he called them little animals. The bacteria and prokaryote is what he discovered. And you guys might remember back to a scientist named Francisco Redi. He did an experiment about spontaneous generation. And basically what they thought back in the 17th, 16th century was that living things could just pop up out of anywhere. So if you left out a piece of meat and maggots grew on it, the maggots just grew out of the meat because you left them there. Or bacteria just grew out of a solution because it was there. So Leeuwenhoek and a couple of other scientists, such as Redi, didn't think this, so they did experiments to prove that that was not right. And so this is one that Louis Pasteur did. Remember, we talked about Pasteur last section too. They had 2 hypotheses. One was that bacteria arise spontaneously in a broth, and the hypothesis B was that bacteria in the air contaminate a broth. So spontaneous generation says that bacteria are just going to grow out of the broth and come from nowhere. And the hypothesis B says that bacteria actually come from the air and contaminate whatever's there. So the first experiment, they had 2 flasks, and they boiled both of them to sterilize them. And in one of them, they opened the flasks briefly outside, and we saw that 89% of them showed bacterial growth. And the other group had boiled the flasks and then opened them briefly inside, and 32% of them showed growth. So in this case, hypothesis B is supported because the relatives of concentration of bacteria in the air explain the results. So there's going to be more bacteria outside than inside. So that is why we saw more bacteria in the ones that were opened outside than the ones that were opened inside. The second experiment had this nifty little setup where the broth was down here, and there was this flask opening right here. And so what would happen is they would boil it in the air and to kill everything, and the bacteria couldn't get all the way back up here. They would get stuck right down here in this little bend. So if bacteria spontaneously generated and we boiled this, they would still grow back in here. We would still find growth, bacterial growth in the broth. However, if they come from the air, then there should be no bacteria in this after we boil it, because the bacteria gets stuck right here and they can't get back up there. And so that's what they found. I know my picture's covering it, but you can look on page 372 in your textbook. 100% of the flasks did not have any bacterial growth, and they were all had bacteria collected in this little divot right here. So hypothesis B was again supported. So they showed that bacteria don't spontaneously generate, they come from the air. So let's talk about the structure of prokaryotes. They are very, very small, about 1 to 10 micrometers in length. That's what that little U means, micrometers. To put this into perspective, as your textbook does, humans are about 1 meter tall, which is about 1,000,000 times longer than a bacteria. So that's pretty small. A meter is 1.0 times 10 to the 10th, whereas micrometers are times 10 to the negative 9. So that's 0.000000, I stopped counting, 8 zeros, 1. And humans are 1 meter tall. Micrometer, bacteria would be 0.00000001. So that's very tiny. They lack membrane-bound nuclei, so you guys know that. Bacteria and archaea are prokaryotes that do not have a nucleus. That's the main difference between prokaryotic cells and eukaryotic cells. Kary refers to the nucleus, and pro means before. So these evolved before a nucleus. They have evolved. They were the first living things on Earth. We have found fossils from about 350 billion years ago. and the fossil record shows that they were alone on Earth for about 1.3 billion years. So they were around for 1.3 billion years before a nucleus evolved by the process of endosymbiosis. Don't forget that. That's how we got eukaryotic cells. So they can live in almost any environment because they are so diverse, and they can capture and use energy in so many different ways. They can be phototrophic or chemiotrophic or heterotrophic. They've basically covered every single possibility. They all have a cell wall outside of the plasma membrane, and many bacteria also have a glycocalyx outside the cell wall. And a glycocalyx is a layer of polysaccharides, which are carbohydrates, a layer of sugars around the cell wall, and there can be a couple of them, a couple different kinds. So if this glycocalyx is very well organized and structured, it's called a capsule. Think of it, it encapsulates it, it's solid, it's well structured. Whereas if it's loosely organized, it's called a slime layer. So think of it as like, it's loosely organized, it's kind of like floating around everywhere, it's kind of slimy. So those are the two different kinds of glycocalyx when it comes to bacteria. This is what many prokaryotic cells look like. They have a couple of different structures I want to point out to you. So one of them is a flagella. So many bacterial or prokaryotic cells have a flagella, which is a bunch of microtubules or a bunch of cytoskeleton that sticks off in our cupboard, and they can rotate or flap back and forth that allow them to have motion, very easy motion. So that is the flagella. Archaea also have them, but they are smaller than those in bacteria. Then they also have these things called fimbriae right here, which are hair-like bristles that allow adhesion to surfaces. So this is how they can stick to things and not just kind of float around all over the place. So this is actually how, if you get gonorrhea, this is how it invades your cells, because it uses the fimbriae to stick onto your cells. That's the example your textbook gives, so be warned of that. They do not have membrane-bound organelles or a membrane-bound nucleus. So they still have DNA. Every living thing has DNA, but they do not have a nucleus. So instead, their DNA is just free inside the cytoplasm. But it is concentrated in one particular spot called the nucleoid. So the bacterial chromosome is in one spot called the nucleoid. It's not bound by a nuclear envelope like inside eukaryotic cells, but it is still there. And remember, the prokaryotic or bacterial chromosome is one single circle. So it's all just condensed up and stuck inside the center of the nucleus. There are also many plasmids, which you should recall are small little circles of DNA floating around that are not necessarily a part of the chromosome, but they are still in there. They have ribosomes, which are important. Obviously, they need proteins. Every living thing needs proteins to do its jobs. So they do have a lot of ribosomes, which are free in the cytoplasm. Anything else on here? They have a plasma membrane, capsule, which can be which is the glycocalyx. It can be a capsule or a slime layer. They have a cell wall, and this picture, which my picture is covering, look in your textbook, shows how a flagella is attached to the cell wall. So it kind of rotates in a 360 degree motion to allow for movement. Oh, and one last thing is the pilus, which we will get to in the next slide, but this is what the conjugation pilus looks like. So here are the different things. A prokaryotic cell has a cell envelope, which is made up of a glycocalyx, cell wall, plasma membrane. The cytoplasm has a nucleoid, not a nucleus, but at one spot where the DNA is. Ribosomes and thylakoids, so those that are photosynthetic need a place to do their photosynthesis, so they have thylakoid membranes. And they also have things hanging off of them. They're appendages, like our arms and legs, except they have flagella, fimbriae, and what is called a conjugation pilus, which we'll talk about. So, reproduction in prokaryotes is obviously different than in eukaryotes. What the process that they use to divide is called binary fission. This is not mitosis, because mitosis is division of the nucleus, and they do not have a nucleus. So, don't be confused about that. It's pretty similar, but it's not the same, because mitosis is the whole process of dividing a nucleus, and they don't have one. So what happens is they have their single circular chromosome. It replicates just like ours does during S phase of mitosis, or the cell cycle. It replicates, and then we have two new daughter chromosomes. They split in half, move to opposite ends of the cell, and the new cell membrane and cell wall form between the two daughter cells. These can reproduce in under 12 minutes. That's pretty crazy. 12 minutes per division allows for very, very, very rapid reproduction, very, very rapid infection if the bacteria is leading to an infection. And because of this, mutations are generated and passed on very, very rapidly. Think about it. A generation time for a human is what? 25, 30, 40, 50 years sometimes. This is 12 minutes of generation time. So think of how quickly mutations in humans can be passed on, which is pretty slowly. But if we were to cut that down from 30 years to 12 minutes, our mutations would happen much more rapidly. And also, the prokaryotes are haploid because they only have one copy of each allele, so mutations are immediately subjected to natural selection. So think of it this way. If you had a mutation that was negative, but you had another allele to cover it up, you had a dominant allele to cover your negative recessive allele, it's not going to get acted on because you're not going to show the phenotype of that negative allele. In this case, there's no dominant alleles to cover your bad recessive alleles, and if you have a bad recessive allele, that prokaryote's not going to live for very long. Now, the reproduction. So what they do is they need to have genetic recombination, obviously. So if they're always reproducing asexually, there would be no changes and there wouldn't be as much evolution. They need to have new genes introduced to them because even though they're mutating really rapidly, they still need to have genetic recombination. So there's a couple of ways. One of them is conjugation. What that means is two bacteria will line up next to each other, use the conjugation pilus, which is by definition how does your textbook define it? It's just a little offshoot of the cell that connects to another one through which they can pass DNA. So basically one bacteria will just donate a piece of its DNA to the other one picks it up, incorporates it into its chromosome, and now that bacteria has that DNA. That's conjugation. Then there's also transformation. And transformation is when a bacteria is just going to come into contact with a small piece of DNA, pick it up, put it into its chromosome. So here we see that a cell has burst, so now there's free DNA in the solution. It burst for whatever reason, but now its DNA is just floating around. This bacteria picks it up and incorporates it into its genome. So now it's a part of that bacterial DNA. It's going to make all of the genes that it's on that DNA that it found. Then the last one is transduction. Transduction is when a bacteriophage, so a virus that only infects bacteria, injects its DNA into the bacteria, and then the bacteria picks it up and uses it as part of its DNA. Now, often this would be an infection, a viral infection, and the bacteria would turn off and become a virus-making factory. But sometimes it's not always an infectious piece of DNA. So in this case, this was a abnormal phage, which has bacterial DNA instead of viral DNA. So it's just an extra piece of bacterial DNA. And that's it. That is the prokaryotes as much as you need to know for now.